So the B cells will be activated. Once the B cells are activated, they go through clonal selection. Clonal selection is a process of dividing the B cells in two groups. One half portion of the B cells will remain as a memory cell. This will remain as a memory cell, and on the subsequent attack of the same antigen, they will get activated. They will multiply and they will produce more number of antibodies against the same antigen. And this is the reason why number of disease are not infecting a person second time in a lifetime. For an example, chicken pox, they will only be repeated once. They will only occur once only during the entire lifetime. The second portion is the circulating B cells. This will impart the immunity. This will produce the antibodies at a present infection, and they are producing multiple clones. Now, as I said, that the B cells which are circulating in our body are synthesizing, are producing different types of antibodies. Okay, there are n number of B cells present in our body. Say this B cell has identified the antigen from a particular surface. There is another B cell which is identifying the antigen from a different surface. Then they are producing the antibodies which are complementary to this structure, and hence our body is mixture of different types of antibodies, against the same antigen. Antigen is having a vast surface. So say this is an antigen molecule and a bacterium. If this surface is red by antigen B cell, the antibodies will be complementary to this structure. They will bind over here. If this structure is red, then the antibodies which are produced will bind with this place. If it is red by this at this place, the antibodies which are produced will bind at this receptor site or this site. So our blood produces different types of antibodies against a same antigen. All will read that antigen, all will bind with that antigen only. But our blood is a mixture of different types of antibodies. If we can isolate a single B cell, a single clone of B cell, once it has gone through clonal selection, as I said, half are as a memory cell. They will not produce antibodies, they will only act as a memory cell. Half will produce the antibodies. If we select any B cell from this producer B cells and we combine it with the carcinoma cells, because they are having slow proliferation, multiplication is slow. So in order to produce more number of antibodies, if we bind this B cells, combine with B cells with any carcinoma cells, cancer cells, they will proliferate faster, they will produce faster, multiply faster, and they will produce more number of antibodies. Now these antibodies which are produced by a single clone of the B cell are called monoclonal antibodies. They will be only single type. Our blood is a mixture, it is having polyclones. This is monoclonal antibody. Okay, there are a number of products available in the market which are based on monoclonal antibodies. Now the antibody can combine specifically with a particular antigen only. So antigen antibody reactions are highly selective in nature. Out of a mixture of antigen present, a particular antibody will bind with its complementary antigen only. And this is the science, this is the logic behind the serological reactions. That is the laboratory test. If you go for typhoid test, it is antigen antibody reaction. If you go for blood grouping, it is antigen antibody reaction. So out of available n number of antigens, the antibody will bind particularly with a single antigen, its complementary antigen. Let us take an example of blood grouping. In blood grouping, what we are doing is, we are taking 3 drops of blood on a slide. We place anti-A, anti-B and anti-RH. So if the RBCs are having the A receptor on their surface, the anti-A will lead to binding, and that will lead to clumping, that is the agglutination. If the blood group is B positive, then B antigen are present on the RBC surface, so anti-B will bind with it, it will lead to agglutination. If Rh factor is present, then clumping will be there. Rh is absent, no clumping will be there. If A and B both are present, then clumping will be observed in both A and B. And if blood group is O, no clumping will happen in A and B. So this is the science of the ABO blood grouping system. This is because of antigen antibody reaction only. We are incorporating anti-A, anti-B and anti-Rh. So they are highly selective. They will bind with a particular antigen only. Same is the case with the uh, antigens, that is virus or bacteria. And that is why we have to provide vaccines for each individual disease. A single vaccine can't work with all the microorganisms. If we are providing TB vaccine, that is a BCG vaccine, it will only provide protection against mycobacterium. If we are giving polio vaccine, it will provide only protection against the polio. Now, there is one exception, that is not exactly exception, but sometimes it may happen that the antigenic similarity is there. Two antigens are having similar structure, and because of that, we are getting protection against a disease due to antigenic similarity. The first vaccine which was discovered, I think last lecture we have discussed that. Which was the first vaccine discovered? Discuss થયું છે કે નથી થયું? થઈ ગયું છે લાસ્ટ લેક્ચરમાં. What was the first vaccine? BCG. વર્લ્ડની ફર્સ્ટ વેક્સિન કઈ હતી? શીતળાની રસી. Okay, that is smallpox vaccine. Who discovered it? Edward Jenner. How it discovered? How it was discovered? Anyone? નહીં. Okay. So એની ડિસ્કવરી કઈ રીતે થઈ છે આપણે ડિસ્કસ. Okay. આપણે ડિસ્કસ નથી થયું ને લેક્ચરમાં ટૂંકમાં. So when there was an outburst of smallpox, and initially Edward Jenner injected the pus from the cowpox blisters to the chicks, the hens. Out of them, few survived, few died. But those who survived were immunized against chickenpox. Then he got the idea that this can also work with the humans, and hence he injected the pus from the blisters of cowpox to the son of his gardener. The kid developed fever, but he didn't develop severe symptoms, and the kid was immunized against the chickenpox as well. That is antigenic similarity. The virus of cowpox and chickenpox are nearly similar. So when Edward Jenner injected the pus which contained the virus of cowpox, it provided protection against chickenpox. Okay, so that is because of antigenic similarity, and hence the vaccine was discovered. The same is used currently with the rotavirus vaccine. Currently available rotavirus vaccine is having only 5 strains of rotavirus, while rotavirus is a large family. It contains I think 17 different strains of virus. All can infect the body. They are responsible for GI problems, particularly in the kids, and they infect the intestinal membrane, lead to severe diarrhea and sometimes even death. This vaccine is included in the universal immunization program of Government of India as well. It is available free of cost at all the PHC and CHCs. Now, this vaccine contains only 5 strains, but all the strains are having different structure, and that is why the immunity which is developed against these 5 strains of the rotavirus will help providing protection against all the 17 strains of the rotavirus. So this is an example of antigenic similarity. So normally the antigen antibody reactions are highly selective in nature, but sometimes due to similar structure of the antigen, they can bind with non-specific antigen as well. Now, the structure of antibody, the antibodies, they are also known as immunoglobulins. The immunoglobulins are having 2 chains. One is heavy, another is light. And there are two pairs of the chains. Let me focus on the structure only. We're not going in much detail over here. So this is the structure of the immunoglobulin. If you look at the structure, there are two long chains. They are called H chains or heavy chains. And there are two light chains or L chains. These heavy chains, they contain approximately 450 amino acids, while the light chains, they contain nearly 230 amino acids. Two heavy chains are joined together through 1 to 2 disulfide bonds. These are disulfide bonds. There are two disulfide bonds present over here. Okay, so two heavy chains are joined together through a disulfide bond. Now, and light chains are attached to heavy chains through disulfide bond, as shown over here. These are the light chains. They are attached to the heavy chain. Now, where there is a disulfide bond, these bonds are slightly flexible, and because the light chains are attached to the heavy chains, over there the structure is somewhat flexible. It can bend and twist. Mind well, both bending and twisting both are possible, and because of that, when the antibodies are present in blood, both these chains are intricately folded with each other. They are folded with each other as shown over here. Similarly, at the tail also they are folded, and the entire structure appears like gamma of Latin or Y, and that is why they are called gamma globulins. Okay, gamma globulins. This is because of the gamma shape, and they are globular proteins. As a whole, the molecule will appear like a sphere, like a globule. That is why they are called gamma globulins. Now, each heavy chain is having the molecular weight of approximately 50,000 daltons, while each light chain is having the molecular weight of approximately 25,000 daltons. In heavy chain as well as in light chain, there are two portions as they are shown over here: VH, CH, VL, CL. VH means the variable portion of the heavy chain, and CL means CH means constant region of the heavy chain. Similarly, VL, that is variable region of the light chain, and CL means constant region of the light chain. Now let me explain what is constant region and what is variable region. As we know that the immunoglobulins are made up of amino acids, they are peptides. Say we have a structure with different amino acids. Okay, I have written an arbitrary structure, or let us assume it. Uh, it's a chain. Okay, number of amino acids are attached together. In a heavy chain, there are 450 amino acids, as I said. In a light chain, there are approximately 230, 225 to 230 amino acids. Out of the total number of amino acids present in a heavy chain or light chain, nearly 50% of the sequence is constant. It is not changing. Okay, so it is like a pizza. When you go to pizza shop, you will order a pizza. They will ask for extra toppings. What you want as a topping, or when you want go to ice cream shop, they will ask for the extra topping. Do you want almonds to be incorporated, or cherry, or anything else as a topping? So the structure of antibody is also like that. The antigen binding site is tailor made, that is specific. Rest of the portion is constant. So nearly 50% of the structure of any antibody is fixed. It is constant. It is not having difference in the sequence of the amino acids. While less than 50% approximately is variable region, in which the amino acids can be arranged in a manner as demanded by the antigen structure. Depending on the structure of antigen, they will have variable structure, variable arrangement of the amino acids. Here, the binding takes place normally by hydrogen bonds or London forces or van der Waals forces. Okay, so these are the forces of weak attraction, and if multiple bonds are formed, then and then only the antigen can bind firmly with an antibody. Single bond won't work, like ionic bond or the covalent bonds. The single bond is sufficient to hold two atoms together. Over here, it is not, and that's why multiple bonding sites should be there, and that is why a complementary structure is to be produced. So, suppose in an amino acid, C double bond O group is there, then it will bind with the NH2 group because of due to hydrogen bonding. Okay, clear? So, two opposite natured group should be exposed to each other in arrangement of the immunoglobulin and antigen. So, whatever structure is present in an antigen, the B cells will produce antibodies which are complementary to that structure. Say, three amino acids which are present on an antigen are having the structure like CO, then there is a space, then NH2, there is a space, and again CO. So the B cell will try to synthesize to produce the structure which is complementary. Over here it will be NH, then space, then CO, then space, and then NH. So it can form multiple hydrogen bonds. Okay, so that portion is variable, that is changing. Now let me write structure as ABCD for an example. So in an amino acid sequence, suppose this portion is constant. Over here there will be jumbling. There will be change in the sequence. So this region is called constant region. This is called variable region. Both are important. Now, the constant region will decide the type of antibody, type of immunoglobulin. There are 5 types of heavy chains available. There are 5 different sequence, constant region sequence are available for heavy chains. And accordingly, the heavy chains are categorized as alpha, gamma, alpha, mu, delta, and epsilon. There are 5 different sequences are available in heavy chains: gamma, alpha, mu, delta, and epsilon, and that will decide the type of antibody, type of immunoglobulin. If the immunoglobulin is having heavy chain with constant portion of gamma, then this is called IgG, immunoglobulin G. If alpha, IgA; if mu, IgM; if delta, IgD; and epsilon for IgE. Simple trick to remember is GAMDE. There are 5 types of antibody, that is immunoglobulin molecules, GAMDE, and they are having 5 different heavy chains. So this structure, for an example, is of IgG. Then IgA will have a different structure over here. as a constant region. Variable is dependent on the antibody. IgE will have another structure. So there are 5 sequences available for heavy chains. Similarly, there are 2 patterns available for light chains, kappa and lambda. Only 2 sequences are available for light chains, kappa and lambda. In any immunoglobulin structure, either kappa chain is present or lambda chain is present. That is, both the chains are either kappa or either lambda. In any molecule of immunoglobulin, both type of chains are never present together. Okay, 2 light chains as they are shown over here. So both will be either kappa or either lambda. Here there is no specific uh, rule that all the kappa will be present in IgG or all the lambda will be present in IgA. That's not an issue. All IgG, A, M, D, E may have either kappa chains or lambda chains, but both together are never present. Okay. So now in the variable region also there are some pockets. Yes. Nah. Nahi, 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 nahi. Okay. Both heavy chains are of same type always. That is deciding the type of immunoglobulin. Both are gamma, then it is IgG. Both are delta, then it is IgD. Both are epsilon, then it is IgE. Both will be same. There is never mismatch. Okay. So both H and both L will be of similar type in a particular antigen antibody molecule. Now, in the variable portion also, uh, there is not a total variation. There are some pockets of relatively constant portions and relatively variable regions. Now, the relatively variable regions are having very high variation or very high change in the structure, and those regions are called hypervariable regions or hot spots. Okay? Hypervariable regions or hot spots. In heavy chain, there are normally 3 to 4 hot spots present. In a light chain, there are normally 2 to 3 hot spots are present. And these hot spots will decide the CDR, that is complementary determining region. Complementary determining region, actual region where the complement binding or the binding of amino acids will take place. That is called CDR, complementary determining region. Normally, this region lies in the upper part of the antibody molecule, as it's shown over here with a dotted box. This is the site at which antigen will bind. As the antibody is a peptide, it's a small molecule. If even smaller antigen is there, then one antibody molecule can bind with 2 antigens at both the ends, both the arms. But this is rarely happening because the smallest antigens which invade the body are the toxins which are produced by the bacteria. They are also enzymes or peptides. So they are larger enough that can cover both the sides. But if smaller molecules are there, then they can one immunoglobulin can bind with 2, or it can bind with 2 sides of the same antigen as well. So one antibody molecule may have 1 or 2 binding sites at the upper region, that is where both L and H chains are present. This region where the 2 heavy chains are joined together with disulfide bond is sometimes also called hinge region. As we are having hinges in our doors and they make the doors help the door in opening and closing. Okay. Similarly, this hinge region of the antibody molecule where both chains are joined together is providing movement, as I said earlier. Twisting and bending both is possible depending on the surface of the antigen. This can twist and bend. Okay, twisting it as a and a bending it as, and that's why that will help in binding with the antigen. Sometimes this portion is also referred to as constant fraction Fc and Fv, variable fraction. Okay, Fv, Fc, fraction, variable fraction and constant fraction, or constant region and variable region. Now, as I said, there are 5 classes of the antigen immunoglobulins present. First is IgG, immunoglobulin G. Immunoglobulin G is the only immunoglobulin that can cross the placenta and that imparts natural passive immunity. Okay, it is the only immunoglobulin that can cross placental barrier, and that is why it can transfer to the womb, and that imparts natural passive immunity. This is the reason why some disease won't infect a kid up to 6 months of the age. So, because of the IgG molecules, this is the most abundant. About 80% of all antibodies in the blood, which are IgG molecules. It is a monomer. It's a single unit structure. IgM is a pentamer. We will discuss later on. Okay. It protects against the bacteria and virus by enhancing phagocytosis, neutralizing toxins, and triggering the complement system. It is the only to cross the placenta, and that imparts the immunity to the offspring, newborns. IgA molecule, it's a dimer. This IgA also is responsible for the passive immunity, naturally acquired passive immunity. IgA is fat soluble immunoglobulin and it is present in colostrum, a yellow color fluid which is secreted along with the breast milk, that is rich with the IgA, and that imparts natural passive immunity to the breastfed kid. That is why the doctors normally prescribe, suggest that the kid should be breastfed up to at least 6 months of the age, because it is rich with the IgA, immunoglobulin A molecules, and that imparts the natural passive immunity to the kids. It is also found in sweat, tear, saliva, mucus secretions, and gastrointestinal secretions. There are smaller quantities are present in the blood and lymph. It make up 10 to 15% of all the antibodies in the blood, and it occurs as a monomer and dimer both. The level decreases. Yeah, this is important. The level decreases during stress, and it leads to the decreased resistance. This is the reason why number of students fell sick during the exams. During exams, they are more prone to infections because they are under stress. In any conditions where you are under stress, there are more chances that you will acquire any infection because of decreased immunity. This is IgM. It is also known as macroglobulin because it's a pentamer. There are 5 antibody molecules joined together. It's a pentamer, and that is why it is also known as macroglobulin because of larger size. It is having highest molecular weight, largest size. It is having 5 to 10 binding sites. It is having 5 to 10 binding sites. Totally, 5 to 10% of all antibodies in the blood are IgM molecule. Now, this is the first antibody which is secreted by the plasma cells in the blood. When any antigen invades the body, the first response is the IgM molecules, and hence this is used for diagnosis of number of disease. If IgM molecules against a particular disease are present in blood, they are suggestive of recent invasion, recent infection. So for diagnosis, we are using, recently you might have read about or heard about the diagnosis of dengue. For that, they are having dengue kit. So that is also analyzing the IgM molecules. The blood plasma anti-A, anti-B antibodies of ABO group system They are also IgM molecules. NTRH, NTA, NTB. They are also IgM molecules. IgD. It is mainly found on the surface of B cells as an antigen receptor. As we have seen the structure of the B cells, B lymphocytes, they are present as a surface and they try to read only the antigen. It occurs as a monomer and it is involved in the activation of the B cell. About 0.2% of all the antibodies are IgD molecules and IgE molecules. They are very less in number, but they are responsible for very severe reactions. They are responsible for all kind of allergic reactions, and though they are having very low content in the blood, less content in the blood, they can produce very severe reactions. Sometimes, if not controlled, may result in the death as well. Okay, so if you develop any anaphylactic reactions, for an example, honeybee bite, it triggers the immune response. That is because of the IgM molecules. All kind of allergic reactions are due to IgM molecules. Sorry, IgE molecules. Now, the antigen antibody actions, they are neutralizing the antigen. I think we already discussed that the antibodies will never kill the antigen. Antibodies will only neutralize the antigen. They will precipitate it, they will isolate it, they will make it immobile, they will make it cross-link, anything. Okay, but they will not never neutralize it. The lower portion of the immunoglobulin molecules, Ig molecules, this, when it is bound with antigen, it acts as, so when it is bound with the antigen, it will act as a flag, and that will help in recognition by the phagocytes. So subsequent action of the antigen antibody binding is phagocytosis. We have discussed in the last lecture. Okay, so the phagocytes will get attracted and they will digest the antigen. So that will act as a flag only. They will not neutralize, they will not kill. They will immobilize the bacteria, agglutination and precipitation. Okay, there are two types of antigen antibody reactions possible. One is agglutination, another is precipitation. Both are taking place because of the antigen antibody reaction. Only difference between them is when your antigen is soluble, then it will lead to precipitation. When the antigen is insoluble, it will lead to agglutination. This precipitation sometimes also referred to as flocculation. When the precipitate remains suspended and they form floccules in a solution in a liquid state, that is called flocculation. So principal difference between this flocculation or precipitation and agglutination is the state of the antigen present in the blood, in the sample. If it is soluble like a toxin, it will get precipitated out. If it is insoluble like a bacteria, it will lead to agglutination. So our blood grouping, ABO blood grouping system is agglutination process, because our RBC are insoluble in blood. They will only get, you will only see clumping, aggregation of the RBC molecule, RBC cells. Okay, they are already insoluble. When it is soluble, you will observe precipitation. Then they will activate the complement. Complements are the proteins which will help in binding with the phagocytes, and they will enhance the phagocytosis process. So these are the structure. Monoclonal antibodies, I think we already discussed, so we are not going in detail with that. Let us start with the next, that is antigen antibody reactions. Uh, Before that, let us discuss the blotting techniques. They are important blotting techniques. Uh, they are used for detection of proteins, DNA, RNA, mainly three: DNA, RNA, and proteins. All can be detected by or analyzed by blotting techniques. There are three types of blotting techniques available: southern blotting, western blotting, and northern blotting. Okay, southern blotting, western blotting, and northern blotting. These three blotting techniques are used. The literal meaning of blotting is absorption by a paper or cloth. We are using blotting paper. If we are having a liquid on a surface, we'll try to absorb it on a paper or cloth. So that is called blotting. The same thing is used over here also. Clear? In the blotting techniques, we are absorbing the DNA or RNA or protein. They will diffuse through the paper and they will migrate, and their prints will be read by the readers, which are specifically designed from them. These are also involving antigen antibody reactions. So, to remember which technique is used for which uh, type of analyte, you need to remember snowdrop. Snowdrop. Southern blotting is used for analyzing DNA. Northern blotting is used for the analyzing of RNA, and Western blotting is used for analyzing proteins. Okay. Original name, which was discovered, is Southern blotting. That is based on the name of a scientist, and then they were given similar names. So Southern blotting is used for DNA, Northern for RNA, and Western blotting for the proteins. O is cancelled out. Now let us discuss the mechanism. How the Southern blot is the analytical technique which is used in biology, immunogenetics, and other molecular methods to detect or identify DNA of interest from a mixture of DNA or a specific base sequence within a strand of DNA. Uh, this blotting techniques are normally used in the criminal detective analytics or to identify a particular DNA. For an example, in the DNA testing as well. So, what we are doing over here is we are having a fragment of DNA. As you know, the DNA is a double stranded structure. We will first isolate, that is annealing. Both the strands will be separated out and they will be multiplied by PCR, polymerase chain reaction. And then we try to read it by a complementary DNA structure. So we are having a reference DNA structure. If it binds with the reference DNA structure, that shows that this DNA is present. If it doesn't bind, then we are sure that it is not present. Okay, we have one fragment which is known to us, and we want to detect the unknown, that is opposite fragment, which can bind with the particular fragment. So that is the logic. That is the science behind the blotting technique. So it was developed by a molecular biologist E.M. Sodan in 1975 for analyzing the related genes in a gene restriction fragment, and thus named as Southern blotting in his honor. Later two are named as such. So the principle, the process involves the transfer of electrophoresis separated DNA fragments to a carrier membrane, which is usually nitrocellulose. It's a membrane, it will only carry. Uh, we will study the process entirely. So there is a sponge, a membrane between them, and only liquid can diffuse through them. That liquid carries the DNA or RNA or protein. In case of southern blotting, we are only carrying the DNA. In case of western and northern blotting, the protein and RNA. So they can diffuse through that membrane, and they are captured on a nitrocellulose membrane at the top. So the top layer is now having the bands of DNA, and when we allow to react with a complementary structure which is radio labeled, complementary structure is radio labeled. So if a complementary structure binds with them, they can be detected on an X-ray plate. So we can find this type of DNA is present. So Usually, nitrocellulose detecting a target fragment by hybridization probe. Hybridization refers to the process of forming double-stranded DNA between a single-stranded DNA probe and a single-stranded target DNA. We are having already a single-stranded probe which can bind with the target DNA, single-stranded target DNA. So, since the probe and target DNA are complementary to each other, the reaction is specific, which aids in the detection of specific DNA fragment. These are the steps involved in the southern blotting. First thing is we are running the electrophoresis on a gel. We are having wells in which we are placing the sample, and then we expose it to electrophoresis. When we apply a charge, depending on the charge present on the DNA or RNA molecule or a protein molecule and its size, they will diffuse, they will migrate from this well, and they will form an entire band, that is separation. Larger molecules will move at a slower pace. The smaller molecules will move at a faster pace. The molecules who are having higher charge will move at a faster pace. Those who are having lower charge will move at a slower pace because of the attraction and repulsion. Both the ends are having opposite charge. So, the charge to weight ratio is ultimately determining the movement, the speed or the velocity of migration of the molecules, and accordingly they will be separated, they will form different bands. So, once we have separated them, because the serum which we are incorporating is a mixture, it is having n number of DNA fragments. We want to detect a particular type of DNA, and that's why we need to separate them first. So, through electrophoresis, we are separating them. Then we are making a number of layers. So, this is your gel through which you have gone through electrophoresis. It is placed over here. Then the plate is filled with a salt solution. The salt solution will migrate, will move because of the concentration gradient. Then over the gel, we are having a sponge. Sorry, at the bottom we are having a sponge, and above that there is a nitrocellulose membrane. And above nitrocellulose membrane, there is paper towel which will absorb the extra liquid. So the structure is like sandwich. Here there is a sponge. Below that there is salt solution. Above that there is cellulose membrane, nitrocellulose membrane. And above that membrane there are paper towels. So as these are dry, sponge can absorb the salt solution. So the salt solution will diffuse through the sponge, it will diffuse through this gel, and then through the membrane. Now, as the membrane is made up of nitrocellulose, it is having very small pore size. Through that pores, DNA, RNA, or protein molecules cannot migrate. Only salt solution can migrate. So, it will lead to accumulation of the DNA or RNA molecules on the membrane. The salt solution will be transferred to this paper towels. They will absorb it. We can replace them if required, if they are saturated. So this migration will continue. The salt solution will migrate through sponge to the gel, to the cellulose membrane, and finally to the paper towel. So this migration continues. Once we have decided the end point, that is nearly all the salt solution has passed or some time exposure time is provided, we will separate, we will remove the paper towels and we will separate the membrane. Now the cellulose nitrate membrane is rich with the different types of DNA molecules because they cannot cross the membrane. It is a semi-permeable membrane. The DNA or RNA molecules or protein molecules cannot cross it, and that is why they are trapped on the cellulose membrane. So the cellulose membrane is now having different bands of DNA as they were present in the electrophoresis. So it is like a lithography or Xerox at present. What we are saying is, લિથોમાં શું થાય, કંકોત્રી બનાવે છે તો લિથો માટે એક પ્લેટ બનાવી નાખશે. The ink can penetrate through that pores only. Rest of the part will be impermeable to the ink. Here we are making difference by selective permeability of the membrane, which will only allow the passage of salt solution and not the RNA DNA molecule. So they will be trapped over there. Now the membrane is having different bands as they are apparent over here. These are the bands of DNA and RNA. Now what we do is we go for hybridization. We are already having a complementary structure of DNA which we want to detect in the sample. Say we are going for DNA testing of a person. So what we do is we isolate DNA from both parent and a kid. Parent DNA is taken as a marker that is radio labeled. It is multiplied by PCR and it is radio labeled. The kid DNA is run as a electrophoresis and then goes through blotting. Okay, only fragments, half of the fragments. Now, if on this membrane, kid DNA is matching with the parent DNA, which is already radio labeled, then it will bind with it and it will remain as a band. Clear? So, the bands will be formed only at a place where a complementary DNA is present. Because the parent DNA which we have incorporated is radio labeled, and if it is complementary to the kid DNA, child DNA, or detect which we want to detect, then and then only binding will take place. Both are single stranded. Getting it? Okay. So as shown over here in a blotting technique, we are running the plates. We are going through electrophoresis, and before that this DNA are single stranded. Now in a detector we have incorporated complementary DNA. Say initially this is your DNA. We go for separation of both the strands. Clear? Now say we want to detect a particular strand. This strand is separated, removed. Now this strand can bind with this strand only. Agree? Say I want to detect a particular type of DNA. I have that DNA with me. So what I do is I radio label this DNA. I include the nucleotide base. In DNA, we are having deoxyribonucleic acid. There are a number of places at which we can go for radio labeling. We can change the molecules and we can radio label it. If we radio label a complementary DNA, this strand, not this strand actually, the strand which we want to detect, and we are including it as a marker in hybridization. We are dipping this plate in a solution containing marked DNA. So this marked DNA will bind with a complementary DNA only. Okay. જ્યારે બાઇન્ડિંગ થશે ને તો જ પ્લેટ ઉપર રહેશે, બાકી તો પ્લેટ ઉપર રહેવાનું જ નથી. હા, એ તો સોલ્યુશનમાંથી આપણે કાઢી લેશું પ્લેટ, એટલે એમાં બાઇન્ડિંગ થયેલું હશે તો જ રહેશે ને, બાકી ક્યાં પ્લેટ ઉપર રહેશે. જે રેડિયો લેબલ છે એ પ્લેટ ઉપર ક્યારે રહેશે? જ્યારે બાઇન્ડ થઈ શકશે ત્યારે રહેશે. પ્લેટ ઉપર બાઇન્ડ, આપણે પ્લેટ ઉપર જ એબ્ઝોર્બ કરાવેલું છે ને. પ્લેટ એટલે કે મેમ્બ્રેન ઉપર, ફિલ્ટર ઉપર બાઇન્ડ કરેલા છે ડીએનએ ને. પાછું આપણે કરીએ એક વખત. આ અહીંયા આ જેલ છે. જેલની અંદર આપણે અલગ અલગ વેલ્સમાં સેમ્પલ્સ મૂક્યા છે. એ સેમ્પલને આપણે ઇલેક્ટ્રોફોરેસિસથી સેપરેટ કરી નાખશું પહેલા. એટલે આની ઉપર બેન્ડ્સ બની ગયા. Depending on the structure, size, electric charge of the DNA fragments, they will separate out. જે ઇલેક્ટ્રોફોરેસિસ છે એ માત્ર સેપરેશન માટે છે, એટલે આખે આખા પ્લેટ્સ ઉપર બેન્ડ બની ગયા. હવે બરોબર છે. હવે આ બધા બેન્ડ્સ બની ગયા. અહીંયા એક એની સાથે સાથે જે લેબલ્ડ માર્કર પણ રાખેલું છે, રેડિયો લેબલ માર્કર પણ રાખેલું છે, બરોબર, એના પણ સેપરેટ બેન્ડ્સ બન્યા છે. આ બધા બેન્ડ જે બન્યા એને આપણે હવે બ્લોટિંગ કરશું. બ્લોટિંગ કરવાથી શું થયું તો કે બધા બેન્ડ આખે આખા શિફ્ટ થઈ ગયા, ટ્રાન્સફર થઈ ગયા ટુ ધ મેમ્બ્રેન. એટલે એની એક ઝેરોક્સ કોપી જેવું થઈ ગયું હવે, બરોબર છે. જે પેલા જેલ ઉપર હતું એ હવે બધું મેમ્બ્રેન ઉપર આવી ગયું. હવે મેમ્બ્રેનની સાથે કોમ્પ્લિમેન્ટરી બાઇન્ડિંગ કરાવશું. જ્યાં બાઇન્ડિંગ થશે ત્યાં બેન્ડ્સ ક્રિએટ થશે, જ્યાં બાઇન્ડિંગ નહીં થાય ત્યાં કંઈ નહીં થાય, બરોબર છે. એટલે કોની સાથે બાઇન્ડ થશે તો કે એને કોમ્પ્લિમેન્ટરી છે ને એ જ બાઇન્ડ થશે, બરોબર. જે આમાં દેખાય છે આ 3 જગ્યાએ બેન્ડ્સ દેખાય છે, તો એનો મતલબ છે કે આ 3 સ્ટ્રક્ચર્સ છે કે જે પ્રેઝન્ટ છે, જે આને સિમિલર છે કોમ્પ્લિમેન્ટરી સ્ટ્રક્ચરના. getting it? હજી ક્વેશ્ચન છે? હા, હાફ પોર્શન છે એ પહેલેથી પ્રેઝન્ટ હશે, પહેલેથી એટલે આપણે રન કરાવીએ છીએ એ જ, બીજો નહીં. બીજો આપણે પછીથી ઇન્ક્લુડ કરીએ છીએ. એક વખત મેમ્બ્રેન સેપરેટ થઈ ગયું પછી ફિલ્ટર ઇઝ soaked in a solution which is containing the radio labeled fragments, counter fragments. કાઉન્ટર ફ્રેગમેન્ટ છે એ રેડિયો લેબલ છે એની સાથે બાઇન્ડ થશે, બરોબર, હાઇબ્રિડાઇઝેશન થશે અને પછી એને આપણે રીડ કરવાનું છે. when we expose it to x-ray frame the radio labeled compound will illuminate, એ રહેશે અને બાકીના બધા બ્લેન્ક થઈ જશે, બરોબર. એટલે જે કોમ્પ્લિમેન્ટરી સ્ટ્રક્ચર છે they are radio labeled. Let me give an example with a chemical reaction. Suppose I want to detect we can radio label મિથેનોલ, બરોબર છે, ઇથેનોલ છે, અને આપણે રેડિયો લેબલ કરી શકીએ. કાર્બન રેડિયો લેબલ થઈ શકે છે, બરોબર, રેડિયો લેબલ કાર્બન અવેલેબલ છે. હવે મારે કોઈક એક એસિડને ડિટેક્ટ કરવું છે કે એસિડ પ્રેઝન્ટ છે કે નહીં. ફોર ધેટ આઈ વિલ ગો ફોર એસ્ટરિફિકેશન, બરોબર છે. હવે મારી પાસે સામે અલગ અલગ સ્ટ્રક્ચર છે, એસિડ છે, આલ્ડીહાઇડ છે, આલ્કોહોલ છે, પછી હેલાઇડ્સ છે, ઓકે. આની સામેના સ્ટ્રક્ચરમાં મારી પાસે CH3 CH2 COOH છે એક, બીજું છે CH3 CH2 CHO, ત્રીજું છે CH3 CH2 Cl, ચોથું છે CH3 CH2 Br, બરોબર. આ કેમિકલ રિએક્શન્સ છે. મારે ડિટેક્ટ કોને કરવું છે? આલ્કોહોલ, સોરી, એસિડને, ખાલી એસિડને ડિટેક્ટ કરવું છે. હવે હું એસ્ટરિફિકેશન પ્રોસેસ કરીશ તો એસ્ટર કોણ બનાવશે આની સાથે? એસિડ. આલ્ડીહાઇડ એસ્ટર નહીં બનાવી શકે, ક્લોરાઇડ નહીં બનાવી શકે, બ્રોમાઇડ નહીં બનાવી શકે, કે બીજું કોઈ નહીં બનાવી શકે, બરોબર છે. એટલે એ સ્ટ્રક્ચરમાંથી શું આઇસોલેટ થયું? એસિડ. હવે જે સ્ટ્રક્ચર બન્યું શું થશે? ધેટ ઇઝ CH3 CH2 COO CH2 CH3, બરોબર. હવે આમાંથી આટલો પોર્શન જે છે, માય મિસ્ટેક, આ બાજુ આવશે, ઓકે, બંને સાઈડ ચાલે, ઓકે. સો આ પોર્શન જે છે ધેટ ઇઝ રેડિયો લેબલ્ડ. આ કાર્બન છે એ રેડિયો લેબલ્ડ હતો, આ ઇઝીલી ડિટેક્ટ થશે હવે, બરોબર છે. Similar kind of principle is also used in RIA, radio immunoassay, in which we are radio labeling the antibodies, બરોબર. એ પણ આપણે ડિસ્કસ કરશું. રેડિયો લેબલ એન્ટીબોડીને રેડિયો લેબલ કરીએ છીએ અને એન્ટીજન એન્ટીબોડી બાઇન્ડિંગ છે ધેટ ઇઝ અ કોમ્પિટેટિવ પ્રોસેસ, બરોબર. એટલે રેડિયો એક્ટિવિટી તમારા એનાલાઇટમાં કેટલી આવે છે ધેટ ઇઝ રિપ્રેઝેન્ટેટિવ ઓફ ધી પ્રેઝન્ટ ક્વોન્ટિટી ઓફ ધી એન્ટીજન ઓર એન્ટીબોડી વિચ એવર વી વોન્ટ ટુ ડિટેક્ટ. અહીંયા પણ એ જ છે. એટલે જે કાઉન્ટર છે, કોમ્પ્લિમેન્ટરી છે, એ રેડિયો લેબલ છે, બરોબર. પ્રોબ છે એ રેડિયો લેબલ છે. અનબાઉન્ડ પ્રોબને આપણે રિમૂવ કરી નાખ્યો. જે બાઉન્ડ પ્રોબ છે એ ખાલી બચ્યો છે, and that will bind only with the complementary DNA. એનું પોતાના જેવું જ જે ફ્રેગમેન્ટ છે, હાફ ફ્રેગમેન્ટ, સિમ્પલ. આને આપણે મેં તમને સમજાવ્યું કે આને બેને છૂટા પાડી દઈએ, this will only bind with this fragment. બીજું ગમે એટલા DNA પ્રેઝન્ટ હશે ને, they will not bind. ક્લિયર? ના, એક જ પર્સન નું થઈ શકશે. હા, કારણ કે તમારી પાસે પ્રોબ જે છે ને એમાં તમે એક જ રાખ્યું હશે, એક કરતા વધારે. હા, જ્યારે મેચિંગ કરીએ ત્યારે બે ઇન્ડિવિડ્યુઅલ જ હશે. સપોઝ હા ઓકે, બાકીના મિક્સચર. હા, હવે જુઓ કોની સાથે મેચ કરાવે. હવે બે રીતે થઈ શકે છે. સાઇટ ઉપરથી મળ્યું છે એને તમે કોમ્પ્લિમેન્ટરી તરીકે લઈ લો, અને બાકી તમને જે 15 સસ્પેક્ટ છે એ 15 સસ્પેક્ટ નું મિક્સચર તરીકે લઈ લો. ચાલશે? તમને જે સાઇટ ઉપરથી મળ્યું છે ધેટ યુ ગો ફોર પીસીઆર. પીસીઆર કરીને એને એમ્પ્લીફાય કરશો. કદાચ કોઈ ક્રિમિનલ સાઇટ ઉપરથી તમને એક જ ડીએનએ મળ્યું છે, સપોઝ સેમ્પલ એ મળ્યું, you go for PCR, you will multiply it number of times, you can produce 1 million copies of this. આ થઈ ગઈ. હવે એને રેડિયો લેબલ કરી નાખો. This is now radio labeled and I have 15 suspects. I will run the samples of 15 suspects. 15 વેલ બનાવી નાખ્યા મેં. 15 વેલમાં અલગ 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 સસ્પેક્ટના બ્લડ સેમ્પલ્સ મૂકી દીધા. આને રન કરાવીશ, બરોબર છે. જ્યાં બેન્ડ જનરેટ થયો that is the prime suspect. જે નથી એમાં બેન્ડ નહીં ફોર્મ થાય, કારણ કે એની સાથે બાઇન્ડ નહીં થઈ શકે. સમજાઈ ગયું? ના, ફ્રેગમેન્ટ જ. ફ્રેગમેન્ટ આખી આખું ડીએનએ તો લાર્જ મોલેક્યુલ થઈ જશે. વી આર ઓનલી ડિટેક્ટિંગ અ ફ્રેગમેન્ટ. આટલા સુધી મેચિંગ થાય છે એટલું જ. સો વી આર ગોઈંગ ફોર ફ્રેગમેન્ટ ઓનલી. જેમાં એક હા હોઈ શકે ઇટ મે બી. એટલે આપણે એને ડીએનએ ને પહેલા ફ્રેગમેન્ટેશન કરશું, એનું આઇસોલેશન પણ કરશું, એટલે સિંગલ સ્ટ્રેન્ડ જ હશે. આખું ડીએનએ પણ નહીં હોય, સિંગલ સ્ટ્રેન્ડ જ હશે. એને કન્વર્ટ કરી નાખશું સિંગલ સ્ટ્રેન્ડેડ ડીએનએ માં, ધેન વી આર રનિંગ. પહેલામાં પણ એમાં પણ સિંગલ સ્ટ્રેન્ડેડ જ હોવું જોઈએ, નહીતર તો એના પોતાના જ બેન્ડ પ્રેઝન્ટ છે એની સાથે જોડાઈ જશે, તો પછી રીડિંગમાં નહીં આવે. બાઇન્ડ શું કામ થાય? બંને એના એક જ ફ્રેગમેન્ટ પ્રેઝન્ટ હશે. આપણને જે જોઈએ છે એનો કોમ્પ્લિમેન્ટરી ઓપોઝિટ જ બને નહીં હોય. એક ડીએનએ માં અલગ હોઈ શકે. જેટલી રન થશે, જેટલા ફ્રેગમેન્ટ થશે એ થશે, રન થઈ શકે. સાઇડ માર્કર છે ને એ એક લેબલ તરીકે કામ આવે છે. ખરેખર ટેસ્ટ બરોબર થઈ છે કે નહીં, અથવા તો તમે એમાંથી કઈ જગ્યાએ બાઇન્ડિંગ થયું છે, એ એક રેફરન્સ તરીકે કામ આવશે ઘણી વખતે. બરોબર છે કે જેમ કે તમે ટીએલસીમાં એમાં આરએફ વેલ્યુ લ્યો છો, તો તમે એને રેફરન્સ સાથે કલેક્ટ કરો છો. બરોબર છે, એટલે એના માટે ઘણી વખતે સાઇડ માર્કર્સ કામ આવશે. આ પણ રેડિયો લેબલ છે. એટલે આ જો આ બેન્ડ જનરેટ ના થાય ને, તો એનો મતલબ છે કે કંઈક પ્રોબ્લેમ થયો છે, તમારું બ્લોટિંગ બરોબર નથી થયું. અહીંયા બધા બેન્ડ થયા છે ફોર્મ, એનો મતલબ છે કે તમારું બ્લોટિંગ બરોબર થયું છે. બરોબર, અહીંયા તમે પહેલાથી જ રેડિયો લેબલ જ નાખ્યું છે, બાકીના તમારા સેમ્પલ છે. એટલે ધીસ પ્રોવાઇડ્સ યુ ધ સ્યોરિટી કે બ્લોટિંગ બરોબર થયું છે. કદાચ આમાંથી એક પણ બેન્ડ ફોર્મ ના થાય તો એનો મતલબ એવો થયો કે બ્લોટિંગ નથી થયું બરોબર. બ્લોટિંગ નથી થયું એટલે કદાચ કોઈ ડીએનએ એવું રહી ગયું હોય કે જે ટ્રાન્સફર ના થયું હોય. એટલે અહીંયા જુઓ બધા બેન્ડ ફોર્મ થયા છે, થવા જ જોઈએ. બરોબર, એટલે આ પહેલાથી રેડિયો લેબલ છે, એનામાં કોઈ બાઇન્ડ નહીં, એની સાથે કોઈ બાઇન્ડ નહીં થાય. એ નોન જ છે, રેડિયો લેબલ છે, પણ એની સાથે કોઈ બાઇન્ડ નહીં થાય. એ પોતે જ રેડિયો લેબલ છે, એટલા માટે તમને પ્લેટમાં એના બેન્ડ્સ બતાવે છે, બાકીનામાં નથી બતાતા. હવે જુઓ બાકીના બધા રેડિયો લેબલ નહોતા, એની સાથે જ્યારે રેડિયો લેબલ ફ્રેગમેન્ટ જોઈન થયો ત્યારે એ રેડિયો લેબલ બન્યા. ત્યારે એને એક્સરેથી એક્સપોઝ કરીએ તો તમને બેન્ડ્સ મળ્યા એના. એટલે આ 3 જગ્યાએ બેન્ડ મળ્યા, એટલે એનો મતલબ છે કે આ 3 ટાઈપના ડીએનએ સાથે મેચ થાય છે, બાકીના સાથે નથી થતું ફ્રેગમેન્ટ. બરોબર, એટલે આ એનું વેલિડેશન માટે છે. ક્લિયર? Now these are the steps. I'll share this PPT in your group, you can read it. So first is extraction and purification, then restriction, separation by electrophoresis, depurination માંથી purines કાઢવાના છે, denaturation, bloating, baking, hybridization, washing of unbound probes. જે વધારે એક્સ્ટ્રા રહી ગયા છે રેડિયો લેબલ એને આપણે વોશ કરી નાખશું, એટલે બાઉન્ડ થયેલા છે એ જ ત્યાં રહેશે, અનબાઉન્ડ બધા નીકળી જશે. and then the auto radiograph, એને રેડિયો એક્સરેથી એક્સપોઝ કરશું એટલે રેડિયોગ્રાફ જનરેટ થશે. This is used for identification of DNA in the length polymorphism, DNA ટેસ્ટિંગ, પછી ફિંગરપ્રિન્ટિંગ માટે, ક્રિમિનલ ડિટેક્શન માટે આ ઘણી બધી જગ્યાએ યુઝ થઈ શકે છે. Western blotting, also known as the immunoblotting, because an antibody is used to specifically detect its antigen. જેમાં આપણે ડિસ્કસ કર્યું કે that is used for proteins. બરોબર, Western blotting is used for protein, so it is used for detect specific antigen. Proteins, antigens are protein molecules. We are detecting proteins only. It was introduced by Tobin in 1979, and it is now routine technique for the protein analysis. સૌથી વધારે abundantly use થતું હોય ત્યારે Western blotting use થાય છે. Western blotting can produce qualitative and semi-quantitative data about the protein of interest. It helps in identification of specific proteins from mixture of proteins which are extracted from cells, as well as evaluation of their size and amount. The process is nearly similar. એમાં કઈ બહુ મોટો difference નથી. Now, the antigen antibody reactions. As we have discussed, that antigen antibody reactions are highly selective in nature. So, when they are performed in vitro outside the body, they are referred to as serological reactions, because we are using the word sera for the antibody containing preparations or serum for blood. So, these reactions are taking place with blood serum. They are known as serological reactions. In vitro antigen antibody reactions, they are also referred to as serological reactions. So there are two types of antigen antibody reactions. When they are taking place in vivo, they are used for diagnosis of the disease like diphtheria and tuberculosis. Sick test is used for diagnosis of diphtheria and tuberculosis. It is done by tuberculin test. These two are in vivo diagnostic tests. Okay? These two are in vivo diagnostic tests. Here, the test is done on the living subject and on the body. So, in tuberculin test, tuberculin, that is a protein from the mycobacterium. When any individual is exposed to mycobacterium, that is, he has developed infection. The infection may be subclinical or clinical. He may or may not show any symptoms, but still his body is highly sensitive to a protein constituent of the mycobacterium, and that protein is tuberculin. So, in tuberculin test, we are isolating that tuberculin protein from the mycobacterium, and we are injecting that tuberculin to a person for which we want to go for diagnosis. Normally, the tuberculins are injected in the forearm over here, okay, through subcutaneous route beneath the skin, and normally 0.1 ml of the tuberculin is injected. (0.1 ml of the tuberculin is injected subcutaneously. After 48 to 72 hours, the response is recorded. Now, the response will be observed as flushing, that is swelling, or redness, or sometimes even necrosis, totally tissue damage, tissue death. So, it will be observed as an entire circle. The diameter of the circle decides the presence or absence of the TB, that is infection or no infection of the TB. As it's a protein, the body will react to a smaller extent. But if the person is suffering from tuberculosis, his body will react in a very exaggerated manner, very extensive manner, and that's why that infected person will develop a larger diameter. The non-infected person will develop a smaller diameter. Now, the diameters are normally read by the physicians using pen. What they do is they run a pen across the injection site. What happens over here is when the skin is swollen, then the skin becomes rough and the pen cannot run through that skin. If we try to run pen through the surface of our grandparents, ઓકે આપણા દાદા દાદી છે એની સ્કિન ઉપર પેન ચલાવવાની ટ્રાય કરજો, સરખી ચાલશે? લખતા લખતા શું થશે, વચ્ચે વચ્ચે તમારી પેન અટકી જશે, તમારી સ્કિન ઉપર ચાલશે. કેમ? તો કે એની સ્કિન છે એ રફ છે, એક ટાઈમે સ્ટ્રેચ થયેલી હતી, હવે વીક પડી ગયા એટલે એની સ્કિન છે એ કરચલી વાળી થઈ ગઈ. ટ્યુબરક્યુલિનમાં પણ એવું થશે, એટલો પોર્શન છે કે જ્યાં પેલા સ્વેલિંગ થયું હતું હવે સ્વેલિંગ કદાચ મટી ગયું હશે ને તો ત્યાંની સ્કિન છે એ કરચલી વાળી હશે એટલે પેન ચાલશે નહીં. એટલે એ ચારેક કોર્નરથી પેન ચલાવવાની સ્ટાર્ટ કરશે, જ્યાં પેન સ્ટોપ થાય ને ત્યાં એ માર્ક કરી નાખશે and they will read the diameter. Normally the diameter less than 5 mm is suggestive of no tuberculosis and diameter more than 15 mm is always suggestive of tuberculosis. Confusion kind? More than 15 mm is always suggestive of tuberculosis. વચ્ચેના જે છે they depends on the conditions. If the person is immunocompromised, suffering from cancer, suffering from HIV, has contacted any person of highly infected area, etc. Depending on that, the results are considered. The serological reactions are in vitro diagnostic. A sick test that is performed for diphtheria. It contains diphtheria toxin and it is also injected in the same manner. But here we are having two things: sick test and sick test control. Both are injected in both different arms, and we read the swelling, the flushing. થાય છે કે નહીં? જો થશે તો બતાવે છે કે ઇમ્યુન જે પર્સન છે that is infected or immune અને otherwise નથી. બરાબર છે ને? diphtheria છે that is normally infecting the body, that is lung, અને મોટા ભાગે તમને બ્રીથનેસનેસ ને એવા બધા થવાના ચાન્સીસ છે. ઇટ ઇઝ પ્રોડ્યુસિંગ એક્ઝોટોક્સિન, બરોબર છે? એટલે એ જે ટોક્સિન્સ છે એ બોડીને વીક કરશે ને એવું બધું થશે. હવે લગભગ ડિપ્થેરિયા બહુ રેરલી જોવા મળે છે, બરોબર? ટીબી ઘણું બધું જોવા મળે છે. ઓકે ઓકે, ચલો એ આપણે ડિસ્કસ કરી લઈએ કઈ રીતે આઈડેન્ટિફાય થાય. અત્યારે આપણે સ્કીપ કરતા હતા પણ વાંધો નહીં. સો જે diphtheria છે ને, sick test, that is composed of two things. One is sick test toxin. This toxin is prepared from the liquid culture. It is a filtrate from the liquid culture of Corynebacterium diphtheria. It is grown on a liquid medium and it is filtrate. So it contains sick test toxin. The dose is adjusted, the solution is made isotonic, and normally the dose is accommodated in 0.1 or 0.2 ml of the solution. So it is accordingly diluted or concentrated. The sick test control is nothing but the toxin which has been heated. That is the filtrate which has been heated at around 60 to 80 degrees centigrade for 1 hour. Sick test control is nothing but the same filtrate which we are taking for the test, but it has been heated to 60 to 80 degrees centigrade for 1 hour. As we know that exotoxins are thermolabile in nature, they can be destroyed. Now the purpose of performing the control test is to eliminate the error due to the cross reactions or the antigenic response due to the growth constituents. Any other constituents which are present in the media in which we are growing the Corynebacterium. It may have some constituents which may be allergic. Okay, it may trigger the allergic response. they may trigger swelling, they may trigger redness. So to separate them, we are preparing control. So control is having everything but the toxin. Toxin has been destroyed by heating at 60 to 80 degrees centigrade. Is it okay? Now, in left arm, we are injecting toxin, and in right arm, we are injecting control. In a left arm, normally we are injecting toxin. In the right arm, same place, we are injecting control. In the same person. Over here, there can be two response. Actually, four combinations, two response. These four possibilities are there. In a right arm, where we have injected the toxin, it may show swelling or no swelling. Swelling, that is flushing, or no flushing. So there are two possibilities over here. Here also there are two possibilities. No reaction or small flushing. Totally four combinations. No, no. Large, no. Small, small. And small, large. Four combinations are there. Now, when there is no response in both, it indicates the person is already having the antitoxin against the diphtheria. His blood is already having the antitoxin against diphtheria. So, he is immune. That is already exposed. If large flushed area over here, no reaction over here, it suggests that he is not having the antitoxin. He is not immune. At present, not infected as well. So, not immune. When small flushed area in both the arms, they are because of pseudo reaction, because of non-specific constituents. There is small flushing in both the arms. So here, there is the person is immune but pseudo reactor. Over here, there is a large flushed area and small flushed area. Not immune and pseudo reactor. So, when we obtain the results 3 and 4, normally it is difficult for us to identify what is small and what is large. In such cases, we need to repeat the observations after 7 days. Readings 3 and 4 are only observed, not repeated, it is observed after 7 days. The pseudo reactions which are present will fade away within 7 days. The true reactions will persist. The reactions due to toxin will be there even after 7 days. The reactions because of the non-specific constituents, the allergic reactions without toxin in the right arm will fade away. So you can easily differentiate that there is no reaction over here now, but still there is reaction over here. So if there is a reaction over there, there is fourth reason. No reaction in both, third one. Clear? So these two observations are to be read again after 7 days. The pseudo reactions will fade away, true reactions will still persist. Clear? Okay. So I think ઇન્ફેક્ટેડ છે કે નહીં, જો એના બોડીમાં ઇન્ફેક્ટેડ હશે તો એના બોડી ટોક્સિન્સ બનાવ્યા હશે, એન્ટીટોક્સિન્સ બનાવ્યા હશે. એન્ટીટોક્સિન બનાવ્યા હશે then એન્ટીટોક્સિન will neutralize the toxin. હા, તો ફર્સ્ટ એ આવી શકે ને થર્ડ એ આવી શકે, આ બંને આવી શકે, સ્યુડો રિએક્ટર હોઈ શકે, એટલે ફર્સ્ટ કે થર્ડ આવશે. જો નહીં હોય તો એન્ટીટોક્સિન નથી બોડીમાં, એન્ટીટોક્સિન નથી એટલે રિસ્પોન્સ આવશે તો સેકન્ડ ને ફોર્થ આવશે. ક્લિયર? So I think we are ending today. Any questions? Anyone? Relay sir is waiting outside. Shall I quickly complete it? Okay. Humoral means humor. Humor means body fluids. Normally, the antibody-mediated immunity is considered as humoral immunity because the antibodies are circulating in blood. They are part of humor body. Okay, and those immunity which is mediated by cells. Normally, the cells which are insoluble, while antibodies are soluble. They can diffuse through liquids. They are present in liquids, and that is why the antibody-mediated immunity is considered as a humoral immunity. Cell-mediated immunity, that is the natural killer cells or phagocytes, all they constitute the cell-mediated immunity. So, in a broader sense, we can consider that natural immunity is considered as it can be considered as a cell-mediated immunity. Then acquired immunity can be considered as a humoral immunity. In a broader term, all the acquired immunity is mediated by antibody only. So, antibody mediated immunity is a humoral immunity. Humor means fluid. Okay. Any other question, anyone? Okay. Autoimmunity is because of the failure of recognition. All our body cells are having receptors, or we can say MHCs. Okay. MHC, that is major histocompatibility complex. Okay, these MHC proteins are present in all the cells of all the individuals. There are mainly 3 types of MHC. We are normally focusing on 2 types at present, MHC1 and MHC2. These MHC1 proteins are present in all cells, while MHC2 are normally present only on the immune cells, that is T cells. Okay, now this MHC1 will make individual we can say immune against all kind of antigens or anything of extraneous material. That is, our cells will protect, our body will protect our own cells. When the person comes in contact with a foreign cell, for an example during organ donation, then the body will recognize that organ as a foreign, and that is why it may lead to organ rejection if the immunosuppressants are not provided, because their MHC proteins are different. And this is the reason why the practitioners also prefer the organs from the blood relatives, so that there are less chances of rejection because their body is having similar kind of MHC proteins on their surface. Okay. Now, autoimmune disease is a disease in which our body fails to recognize our own cells. For an example, a simplest one is the alopecia areata. એલોપેસિયા એરિયાટા, આપણે ગુજરાતીમાં જેને કહીએ છીએ ઉંદરી, બરોબર છે. એમાં શું થાય, આ સિમ્પલેસ્ટ ઓટોઇમ્યુન ડિઝીઝ છે, કોઈ બહુ સિવિયર નથી. સિવિયર નથી એટલે કે ફિઝિયોલોજીકલી સિવિયર નથી, કોસ્મેટિકોલોજીકલી કહીએ તો સિવિયર કહેવાય. ઇટ લીડ્સ ટુ ફોર્મેશન ઓફ પેચીસ ઓફ હેરલેસ ઓર રિજન્સ ઓન યોર બોડી. બરોબર છે, માથામાં કે દાઢીમાં કે મૂછમાં ગમે ત્યાં અમુક પેચમાં વાળ નહીં આવે. That is because our own body fails to recognize our own cells. It treats them as a foreign. And that is why they kill those cells which are producing the hairs, hair follicles. They destroy them. And that's why there are no hair growth over there. This may even spread to the alopecia totalis. બધા જ વાળ પડી જાય અથવા તો બોડીના બધા જ વાળ પડી જાય. That is called alopecia universalis. All are types of autoimmune disease. Another example of the autoimmune disease is Guillain-Barré syndrome, GBS, which I think surfaced more in last 3 or 4 years. After COVID, we have come to know about number of diseases of GBS. Here also, after vaccination, the immune system is triggered and sometimes it is misguided because of the immune response, because at an older age it is difficult to develop the immunity, and this is the reason why we are providing nearly all the vaccines in the younger age, less than 10 years, maximum less than 5 years. Okay, now because of the COVID vaccine, in number of cases the persons who are highly sensitive, they develop GBS. Their immune system was triggered to an extent that it forgot that which is self and which is non-self, and that is why it leads to degeneration of the nerves of number of people. One of our student also died because of GBS 2 years back, who is completed BPharm from this college only. He was qualified GPAT and was going to get admission in MPharm, but he suffered from GBS, he died. So that is another example of the autoimmune disease where the body forgets to recognize between self and non-self. Major because of the MHC. This is the molecule which is helping in recognition. Okay, so any other question, anyone? Guillain-Barré syndrome, Guillain-Barré syndrome, GBS. It's a severe kind of disease. I think it may have taken number of lives after COVID. If it is not controlled in time, it's a life-threatening disease. It's a neurodegenerative disease. Our own immune cells will digest the neurons. Ultimately, leads to death. Any other question, anyone? Okay. ના 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 ના, ઇન્જરીમાં આવવાનું બંધ થઈ જાય છે એનું રીઝન છે કે ત્યાંની સ્કિન જે છે, હવે સ્કિનના આઉટર લેયર્સ છે ને એની અંદર જ હેર ફોલિકલ્સ છે. હવે એ લેયર જો નીકળી ગયું, નોટ આઉટર મોસ્ટ બટ આઉટર લેયર ઉપરનું, હવે અમુક ડેપ્થ સુધી નીકળી ગયું તો ત્યાંથી હેર ફોલિકલ્સ વાળો આખો રીજન જ નીકળી ગયો છે. નવી જે સ્કિન ડેવલપ થશે ને એમાં એ નહીં આવે, એન્ડ ધેટ ઇઝ વાય હેર વિલ નોટ ગ્રો. બરોબર છે, એટલે એવું થઈ શકે કે જ્યાં ઇન્જરી થઈ હોય ત્યાં, પણ ધેટ ઇઝ નોટ ઇમ્યુન ડિસીઝ. ને જે ઇન્જરીના કારણે થયું છે ને સ્પ્રેડ નહીં થાય. બરોબર છે, આ સ્પ્રેડ થશે. આ એક જગ્યાએ થયું છે, પછી થોડાક ટાઈમ પછી બીજી જગ્યાએ પણ થાય, થોડાક ટાઈમ પછી ત્રીજી જગ્યાએ પણ થાય, because the cells can migrate and that's why they may infect another portion. બાકી નહીં. Any other question, anyone? Okay, obviously રહેશે. બરોબર છે. Now when there is organ transplant, the immunosuppressions are provided. At the same time, the persons are advised to take total precautions, stay away from the areas with largely populated or infected, don't visit your relatives. Normally this treatment is ranging between 3 to 6 months only. Till that time the body will now be actually trained to accept that organ when organ donation is there. In alopecia also the same thing happens. Alopecia areata also sometimes the practitioners are prescribing the immunosuppressants like I think one of them is allopurinol, I think. Yeah, that they are prescribing for as an immunosuppressant, and they will train the body for 3 to 6 months. After that, again they will recognize કે હવે આ જે છે એ બરોબર છે આપણું જ છે and it will not fight with them. But for some time it is required to suppress that. The immunosuppressions are also specific sometimes. Which cell you want to target? If you want to target T cells, you will give kind of immunosuppressions will only suppress the T cells. If you want to suppress the entire body immunity, normally steroids are preferred. Okay, like betamethasone. They are generalized immunosuppressants. Any other question, anyone? Clear? Okay then, thank you.